With an understanding of the basic pattern of a blood vessel, which was covered in an earlier presentation, and which is briefly reviewed here, we see that blood vessels have three tunics or layers, as is common with all tubular anatomy. However, there are many named types of blood vessels. Why all the different named vessels, and how do they relate to the basic pattern? In this presentation, you will learn about the smallest of blood vessels, the capillaries, and how and why their anatomy differs from the basic pattern. Recall that capillaries are the smallest blood vessels, having diameters between 5 and 10 micrometers, which approximates the size of a red blood cell. These small tubes, approximately 20 billion in number, form an extensive branching network within the tissues of the body. It is here at the capillaries that exchanges occur between the capillary blood and the interstitial fluid that surrounds the cells. This exchange brings nutrients to the cells, removes metabolic waste produced by the cells, and maintains the water environment that surrounds the body's cells. For this exchange to happen, the capillaries show the most extensive modifications to the basic pattern of blood vessels. Capillaries do not have a tunica externa or a tunica media, and they also lose the internal elastic membrane of the tunica intima. As a result, the blood in the capillaries is separated from the interstitial fluid by the thinnest of cells, a simple squamous endothelial cell with a thin basement membrane which makes diffusional exchanges possible. There are three types of capillaries in the body: continuous capillaries, fenestrated capillaries, and sinusoids. Continuous capillaries are the most common type of capillary. They have plasma membranes that form continuous tubes that are interrupted only by intercellular clefts, thin gaps between neighboring endothelial cells. This type of capillary is found in the central nervous tissue, lungs, skin, skeletal muscle, smooth muscle, and connective tissue. Fenestrated capillaries have intercellular clefts between neighboring endothelial cells and also have small pores called fenestrations in the plasma membrane. This makes these capillaries more permeable than the continuous capillaries. Fenestrated capillaries are found in the kidneys, villi of the small intestine, choroid plexuses of the brain, ciliary processes of the eyes, and most endocrine glands. Sinusoids are the largest and most permeable of the capillaries. They have large fenestrations in their plasma membranes, large intercellular clefts between neighboring endothelial cells, and incomplete basement membranes. These large openings in their wall allow large proteins and even cells, which cannot move through the other capillaries, to enter and leave these capillaries. Sinusoids are found in red bone marrow, the liver, spleen, anterior pituitary, the parathyroid gland, and adrenal gland. We see then that the structure of capillaries facilitates their function as the exchange vessels of the body. However, in a closed circulatory system driven by a pump, a problem is encountered at the microcirculatory level of the capillaries. The net exchange of water is not equal, as more of the water in the blood is moved out of the capillaries than is returned to the capillaries from the interstitial fluid. This problem arises as the pumping heart creates hydrostatic pressure in the closed circulatory vessels, which is not balanced by an equal hydrostatic pressure in the interstitial tissues of the body. While other types of pressure counter the hydrostatic pressure inside the capillaries, they are not as strong as the hydrostatic pressure, and the net effect is a greater movement of water out of the capillaries compared to the return movement of water back into the capillaries. This leads to an increase in interstitial fluid and a decrease in the volume of the blood plasma. This then results in a dangerous problem that needs a solution. In summary, the modifications of the vessel pattern that account for capillary structure allow these small vessels to be well suited for their function as the exchange vessels of the circulatory system. These small 5 to 10 micrometer tubes, numbering approximately 20 billion, form an extensive network of short, branched, interconnecting vessels that course among and make contact with the individual cells of the body to provide them with fuel and remove metabolic wastes. But while maintaining the water environment around the cells, they leave excess water behind during capillary filtration, which reduces the volume of blood plasma returning to the heart. This is a problem that will be solved in the presentation on lymphatic vessels.